Hello everybody, here's Mr. C. This is a screencast for Lab 321 about photosynthesis. My intent here is to give you like a demo of what to do, so you're not just faced with the instructions, but you can watch that short screencast and get you going. Let me say, so make it a little smaller, save some space here on the screen. Here we go. I hope you like my background. It has to do with the lab, kind of, right? We have light here. We have light here. We have water there, and we only thing we need is some carbon dioxide, and we're in business. Here, you're going to study the rate of photosynthesis. So how fast does photosynthesis happen, and we're going to measure that. And you can read through the goals and through the procedure here. This photosynthetic equation is important to understand, and we're going to use it both ways in your answers here. You find in answer 7, you might also think about, hey, what happens if you go from the right to the left in cellular respiration? The lab itself is not really a virtual lab, but you should watch that video here. It's an instructional video by K12. It's going to look like this. It gives you all the background of the lab. Um, that can be very helpful in understanding the lab. Okay, I don't think you hear the sound here, but you're going to watch that short video here, and then I give you even the results of the experiment. To do this experiment in real life, it's hard, it's challenging, especially you would get tired if you do 10 trials. So here's the data for 10 trials where we measure the number of bubbles produced on an Elodea stalk at two temperatures, at a colder temperature and a warmer temperature. Okay, it's not hot, it's warm. And we're gonna, for each of these trials, we recorded how many bubbles of oxygen came out of that stalk magically, right? But it's not a magic if we understand that photosynthetic equation up there. And here are the numbers, and for both of them, cold and warm. And what you have to do in the first part is making a graph. And again, you can do it on paper, but I encourage you to learn how to do this using a spreadsheet like Excel or OpenOffice Sheets or Google Sheets, and do a cut and paste job. If you do it by hand, you can take a picture or scan it in and then insert it as a picture in your space down here. So then we're going to do this, and you answer the questions by typing questions 2 to 7 by typing directly onto this Word document. So those questions are further down. You're only going to submit one. Don't send me a picture and the doc file. It makes things more complicated to grade. So up down here, you need to put in a scatter graph showing the data from both trials, and that graph will have two y-axes, right? We're going to have the trial number on the x, and we're going to have on the first line or first set of data points, we're going to have, actually, I don't want you to create a line. We have the colder temperature, and the second one will be the warmer temperature. So let's just create this. We select the data here. We do a control copy, or do a right click, right mouse click, and say copy. We go to our friend, and I used Excel here. Oops, the background changed. Look at that. Magically. And I create a new spreadsheet here. We're not disturbed. I like different backgrounds. Hope you like that too. We pasted it in, Control V, or you go up here and say paste, and you have various options, right? All you have to do is, at this point, is select, not from beginning from A1, select from A2 all the data. Okay, I hold down the mouse and swipe over it and let go of it. And then I say insert, and in this case, it doesn't really matter whether we use a line graph or a scatter graph, but I like to get you to use scatter graphs because they're more the scientific way. These line graphs are tend to be more business type ways of graphing. So all we need is this type of graph where we have the data points. You need to do some things there where you get the points really in this line item. That is, we want to have also titles for the x-axis, and we want to add a title for the y-axis. And I'll let you figure out what you need to write here. Okay, I want the correct description of what's graphed here on the x-axis, and I think you can find that easily from the table here. And then the same on the y-axis. Okay, tell me what these numbers mean. Give it a short title, and then you grab the chart and control copy or right click and say copy, and you go back to your document with the table already in there. But we go a little bit further down here, right above where it says figure 1, control V. Short note on this is customary in science. We call the graphs figures. We start with 1 and go on, and then we can give this call a caption, some more description. Okay, I put it very cryptic here. You might be a bit longer. Typically, it has more details than the chart title. Okay, but that's all you need to do there, right? If you show me the X and the Y labels with units, okay, these have units. Especially the y-axis has units. The x-axis is the number of trials. Okay, you show me the title and the legend. I'm happy as a baby. Get your 5 points. Okay, then you start working. The first one is, yes, we keep on working on key terms. So explain photosynthesis. I mean, this is the shortest statement you can find here, but it's probably the most work. I want you to write me a paragraph about photosynthesis, explain all parts of the chemical reaction. You have it up there. Explain to me, you know, C6H12O6, what is it? We gave it a name, right? You know, under what conditions does this reaction happen? And so explain to me reactants and products and what else is needed to that it can happen, and always define your key terms. So do a solid job there. I guarantee you it smells like a test question too. You always have to be ready to explain both photosynthesis and cellular respiration. That's pretty easy. Write a conclusion statement of the following question: what is the effect of the temperature on the rate of photosynthesis? So you go back up here and you're going to probably say as shown in figure 1, okay, and now you can talk about things, right? The about average number of bubbles produced here in the colder temperature is about 8 bubbles, I would estimate, and this is about 25 bubbles, right, on average. You can use this as a way to refer to the data. You don't need to refer to each single data point, right? But use these two to make a conclusion statement. Does the temperature have an effect on the rate of photosynthesis? I let it stand like that. I think define photosynthesis very briefly, okay? You don't need to go as detailed as you were up here, okay? Okay, but what is it? Okay, when you use the word here, explain it to me shortly what that is, okay? Then analysis question. Baking soda was dissolved in the water and released carbon dioxide. So it's not just the stock of Eludea in water, there's also baking soda in there. Why is that important? Why is it important to add baking soda? What does it do, baking soda? It's very often used in cooking to create carbon dioxide. Okay, so look at that. There's this one necessary ingredient here is carbon dioxide. Okay, so tell me, you know, this in a nice sentence, and you're gonna get your 3 points there. What advantages did using an Elodea stalk have over using its leaf or leaves? Think about in practical terms. If you have a leaf and this reaction goes on, is it easy to count those bubbles versus on a stalk, right? You look at one stick basically versus a leaf or multiple leaves. It's probably very hard to do, right? And there's another reason, probably the number of cells in a stalk is probably higher than the ones in the leaf or leaves. So make me a nice sentence out of these things. Can you think of a situation where a high temperature would not result in an increase in photosynthesis? So you have to think back, right? Here we obviously saw a higher temperature, a warmer temperature increased photosynthesis. But photosynthesis has more things, more requirements. Look at this. If we anytime limit the amount of CO2, limit the amount of water, limit the amount of light, we're also going to affect the rate of photosynthesis. So think of conditions where we're going to limit light. Okay, you know, does it grow well when it's dark? Does it grow well when it's not humid? Okay, so that should help you with answering that question number 6. And then number 7, if the baking soda had not been added, would there still be a source of carbon dioxide introduced into the water? So we're not talking about traces of carbon dioxide that's already present in the water. Is there any other source of carbon dioxide that we can leverage to make photosynthesis going? And here's the hint. Plants also do cellular respiration. Okay, look at this equation up here again, and cellular respiration is like the opposite of photosynthesis. So if this is photosynthesis, cellular respiration goes from the right to the left. And look what's coming out there. CO2. Okay, and that can help us, right? So explain that and in a bunch of, well, one or two sentences, right? Answer in complete sentences, please. Spend the little time that it takes to get rid of the wiggly lines and all do a spell check, and you get all the points there. I think it's not a hard lab. You should be able to get your 25 points. Some more practice on graphing, and we'll need it. If you do any sort of graphing in the future, it's worth spending the time learning how to do it on the computer, and you're going to be fine. And we're going to have direct instruction on Wednesday at 1 o'clock on the lab too. So in case you want to get going, this screencast should be able to get you going. Where am I? And I hope it's helpful. And otherwise, please send me a K-mail and I'll fix things or answer questions. And I'm always excited to have questions and be able to answer questions for my students. Take care and have fun with the lab.